Hi, I'm Aman.
And hi, I'm Amulia. We're building Novaflow, an AI data analyst for biology labs.
I used to be a computational biologist, and my lab, like many others, always had a large backlog of data that was waiting to be analyzed. I also would often see my colleagues have to wait weeks or even months for the results of their experiments simply because they didn't know how to code. That's why we built Novaflow. I'm going to show you an example of a user using Novaflow in order to do a complex biological data analysis in minutes instead of the weeks that it would normally take. In this case, we're looking at mouse kidney data in a spatial transcriptomics experiment. So Novaflow can automate your entire data processing workflow all the way from your raw data to the visualizations. We're going to be showing you specifically how we take process data and visualize and analyze it in real time. To do that, all you have to do is click Start New Workflow, select your experiment type, and then enter a name and description for this experiment. Then what you'll do is upload the files that have already been processed, either with the Novaflow processing pipeline or that you've processed yourself, and simply you can hit Start Workflow to start processing the files. The AI will then determine the best type of visualization and analysis that you need and run that code automatically. Since this is spatial transcriptomics, we're going to be doing cell clustering, annotation, segmentation, and mapping back to the original tissue. In this case, you can first see the QC metrics popped up so that you can understand how much data that you have and how well your experiment actually worked. Then you can move down to some of the standardized plots. So in this case, we're looking at an image of the tissue taken by the Xenium scanner. Then you can also see how the analysis mapped the UMI counts back to the tissue itself and how the analysis was performed in this animation. You can also see specifically where the analysis area actually was, and when we remove just the tissue, how much of the analysis capture area overlaps with tissue. Then we can look at the cell clustering analysis that was performed, first in 2D space in the standard UMAP methodology, and then we map it back to the actual tissue itself. So you can see where these clusters of cells fall next to each other in a spatially resolved manner. You can also look at different magnifications for this analysis and the clustering. So you can see if certain magnifications, other cell types pop up that might not pop up in smaller magnifications in order to identify rarer cell types. This is just one way you can automate analysis with Novaflow, but we also support many other workflows as well.
Our mission is to put analysis directly back into the hands of scientists, so no matter what kind of experiment you're running, feel free to try us out.
We hope that with Novaflow, you can take your science to the next level.